Types of the mutant viruses. There are different types for the mutant and mutant viruses. Biochemical markers. We can use the biochemical markers for the mutant viruses. Then the deletions. We can delete some specific part of some specific genome or some specific gene, because just like in these days molecular biological point of view studies, we are using some experiments on the eye. We already know that which kind of the gene is already responsible, where is the sequence. We can use the restriction enzymes. We can just put that sequence and we can use restriction enzymes and we can just delete that specific part which is responsible for that disease of the eye, and then we can just use it, and just like this we can delete them. Then the host range is very important, nonsense type of the mutants, and the plaque morphology. There are the temperature sensitive of the mutants. Sometimes the mutants are very sensitive to the temperature, sometimes they are sensitive to the cold. Maybe the temperature sensitivity is the same. Sometimes they are sensitive with the high temperature, sometimes they are sensitive with the low temperature. So reverents, we will see them one by one. First of all are the biochemical markers. Drug resistance mutations. From the drug resistance point of view, it is very important, because I think it was 1980 or about 1980 when the last antibiotic was produced. After that there is no antibiotic. So most of the human beings are resistant to the drugs. The new drugs are not produced as so rapidly. Similarly, the penicillin, whenever we go to the hospital, before using any kind of the injections, doctor always asks us to whether you are the penicillin resistant or not. Why? Because the human beings are really resistant to the penicillins these days, because it has been being used, first generation antibiotic, it has been being used from the decades. Similarly, there are no invention of the new antibiotics, new drugs. So most of the people are being resistant one by one to the already antibiotics available now. So drug resistance mutations are we use the biochemical barriers, altered virulence, even in the molecular cloning, altered virulence, polymorphisms, altered proteins or nucleic acid, we use the biochemical markers, and sensitivity to inactivating agents, biochemical markers are used. Types of the mutant viruses, similar to the nonsense mutations, we are using the biochemical markers. We use the mutant nonsense mutations. It may include one or more of the viral genes and involve non-coding regions of the genome. Again, the similar, just like we need to use the biochemical markers. Defective interfering particles, and it can also revert to the wild type by the recombination by the biochemical markers. Then the host range is very important. Which kind of viruses have which kind of host which we can use for which kind of the host. Just like some viruses are just specific to the chickens. Some viruses will only cause the disease in the human beings. They will not cause the disease in other animals. Some viruses are only causing the disease in the mammals. So this is the host range. How big the host range of some specific viruses refers to the whole host animal or cell type in the vitro. Here are the conditional mutants. So it is especially dependent upon the host of the viruses, whether some viruses can cause the disease in human beings, in the animals, then the animals in the mammals, in the birds. So this is also dependent upon the host range. Nonsense alteration of the coding sequence of 1 or 3 stop codons. As from the pictures given below, you know what is the codon and what is the anticodon. So UAG, UA, and uracil, guanine, and adenine, this is the one codon. So if some nonsense mutation is occurring, it alteration the coding of the sequence of 1 of the 3 stop codons, it will be called as nonsense mutation, because it will just stop it. Termination of the translation will occurs. Low reversion of the frequency for such kind of mutations. Plaque morphology, large plaque mutants or the small plaque mutants are there. The plaque size refer to the temperature sensitivity. As we have already studied, there are some of them are the temperature is very important as far as the mutations are concerned. So if they are sensitive to the low temperature, they are sensitive to the cold. We will see them. The temperature sensitive, useful for the isolation of conditional or lethal mutations. Usually occur by missense mutation in the proteins, and some activities work on the non-permissive temperatures. When the temperatures are very high, the mutations or the replication process is, even in the PCR, we are using some replication processes and it is interacting with the special kind of temperature. When temperature is altered, the results of the PCR are not so ideal as we have thought about them. Then the cold sensitive, opposite to the temperature sensitive, useful in the bacteriophages and plant viruses as their cells can be propagated even at the low temperatures too. Then the reverse. Valid type of the mutations is the reverse. Reverse mutations they are also called as back mutations or compensatory mutations are also there. Sometimes there is some kind of error happens, then it need to be back mutations, and then that back mutation will act upon on that special kind of mutation to make it in the normal way.